want to switch gears a little bit. We've heard a lot of really fascinating work on auditory cortical function at multiple levels, from human indirect recordings to human direct recordings to animal work. And I want to talk a little bit about the anatomy in the human brain. What underlies this interesting functional variation that we see across tasks and across stimuli? It's such a delight to be able to share our anatomical work with the auditory cortex crowd. It's fun to get your abstract picked to give these short talks. And the distinction here, though, really belongs to my graduate student, Ino Liu, who has done the real intellectual and quantitative force behind these ideas. Ino is in the United States on a student visa, and we thought it might be best if she didn't try to cross the border both ways, which is why she's not here to receive this distinction herself. I think it's kind of a testament to the challenges that we face with access at in-person meetings like this one. I hope you get the chance to come stateside and meet her. She's brilliant and her work is extraordinary. If you look at textbook figures of human auditory cortex, it's situated on Heschel's gyrus, abbreviated HG. They're always very clean, they're always very easy to identify. HG is a beautiful prominence. Behind it is a long, flat part of the brain called planum temporale. And if you look at structural atlases that we use in neuroimaging, like FS average from FreeSurfer, also Heschel's gyrus is evident prominently, planum temporale is evident prominently. Even an undergraduate in Introduction to Brain Science, oh, that was mean. Even an undergraduate in Introduction to Brain Sciences can pick them out with some training. But every human brain is unique, and I mean that not in just a touchy-feely way because we're all individually unique, but the anatomy of the human brain is spectacularly variable from person to person. If we digitally dissect the superior temporal plane and look down on it, so we've taken the frontal lobe off, we've taken the middle part of the brain off, you can see that Heschel's gyrus and planum temporale are just not so clear. There's a lot of folding going on the superior temporal plane, and it really makes you wonder what structural landmarks, if any, tell you where primary auditory cortex is in human beings. Classically, neuroanatomists have grouped the variation in the human superior temporal plane into three broad categories: a single Heschel's gyrus that runs medial-laterally along the superior temporal plane, a common stem duplication where you start with one gyrus kind of in the depth of the Sylvian fissure that breaks apart into two like a Y shape, and a complete posterior duplication where you have two Heschel's gyri. He might be the only person to get two gyri labeled for him in the brain, two Heschel's gyri that run the full mediolateral extent. But even knowing these categories, if you look at individual variation, it's tricky. And we would like to be able to say something about the relationship between structural variation and functional variation or behavioral variation. And these variable morphotypes have been associated with a variety of different kinds of cognitive and behavioral phenomena, sometimes in weird and discrepant ways. So multiple gyrification has been associated with communication disorders like dyslexia, DLD, and autism, but multiple gyrification has also been associated with advanced phonetic expertise or musicianship, and it seems like it can't be both. So looking then at individual brains, the patterns are also very hard to identify. Where's the line between a common stem duplication and a complete posterior duplication? How much of a sulcus needs to be present? Does it have to be continuous to be a common stem? It's also extremely tedious to go through and label these by hand, or to train students to become expert anatomists to find these patterns in individual brains. So we'd like to have a way to do this automatically, and then also answer some theoretical questions about auditory cortex. Are these three morphotypes really representative in the data, or are they just something that neuroanatomists have kind of coalesced on over time and then it's become fixed in the literature? Can we link these patterns of macrostructural variation to patterns of microstructural variation or functional variation, and then can we build better alignment algorithms to register different brains to each other for group analyses? So we started with about 1,000 individual curvature patterns we got from the Human Connectome project, which is a remarkable resource. It was fun to see a lot of other HCP posters downstairs this morning. We perform sort of a coarse k-means clustering on the feature matrix, which is based on the curvature at every vertex. We identify the optimal number of clusters to account for variation across individuals in their curvature patterns. And then we want to characterize the morphological, microstructural, and functional features of these different curvature patterns. So what did we find? Good news, there is a single Heschel's gyrus with a planum temporale behind it that emerges prominently from the data. That's nice. We also see the common stem duplication where you have one gyrus that branches into two. We also see the complete posterior duplication where you have two complete gyri that run the mediolateral extent of the superior temporal plane. But interestingly, the clustering algorithm returned another single Heschel's gyrus, and you're like, okay, there are two. Why are there two single Heschel's gyrus? But they differ in a variety of interesting ways. One, the single Heschel's gyrus in orange has a gyrified planum temporale. There's a lot of curvature there compared to the single Heschel's gyrus with a flat planum temporale. There's also a difference in the angle of the gyrus. One runs much more anterior posterior, and the other one runs more mediolateral. Finally, the clustering algorithm returned a fifth category, and I can't tell you, I was delighted to see this one, even though I didn't expect it, which is a single Heschel's gyrus with a notch in it. And when we've been labeling these by hands, we see this one again and again, and you just like, when you see it, you're just like, is it a single gyrus? Is it a common stem duplication? Why doesn't the sulcus run the whole way? It's existentially challenging to identify this gyrus. And it's nice to think of it as its own category. And if you look at the individual brains, and if you come by the poster, I have lots of individual examples for you. If you look at the individual brains, you see that one just again and again and again. So it's interesting that it's its own category. The distribution across the hemispheres is interesting because if you have one kind of folding in one hemisphere, you don't have the same kind of folding in the other hemisphere. Interestingly, the more complex folding patterns, the complete posterior duplication, the common stem duplication, tend to occur more in the right hemisphere than in the left hemisphere. If we characterize these gyri in terms of the macroarchitecture, things like cortical thickness don't vary much across them, but things like the curvature varies quite a bit. So of course, more gyrification means more total curvature, but interestingly, the more curvature you have, the less total surface area you have. And this is sort of, one wonders if the amount of gyrification is helping account for an overall smaller brain or an overall smaller kind of cranial volume. We have work to do here. The heritability is interesting. If you have a common stem duplication, does your twin have a common stem duplication? Does your brother? It turns out that more likely than chance, two related individuals will share these morphotypes compared to two unrelated individuals. So there's a lot of, there's a genetic component here that remains to be identified. In addition to the macrostructure, we'd like to know more about the microstructure because that's what really defines auditory cortex. Microstructure is challenging to measure in vivo because we can't see the cells, but we can see correlates in the MRI signal of kind of cellular populations, including how much myelin there is in the cortex. And this is evident through, for instance, the T1-T2 ratio, which can tell us about intracortical myelination. Intracortical myelination is very high, where you have a lot of heavily myelinated thalamocortical fibers arriving in the cortex, like coniocellular cortex, like primary sensory cortex. And so, of course, primary auditory cortex pops out in the T1-T2 ratio. If we lay that over the different morphotypes on the group average, we can see that where primary auditory cortex might be in these different folding patterns. And if you come by the poster, we have some individual examples too. So the group averages are nice and clean, but again, individual brains are quite unique from one another. We can also functionally characterize these regions. We looked at clusters in the resting state data, voxels that tend to have the same pattern of activity over time. And what's really beautiful about this is the extent to which these clusters of functionally coherent data line up to the different patterns of folding. So we get this one cluster on Heschel's gyrus, one on lateral planum temporale, one on posterior planum temporale that approaches the operculum. In sum, there might be 5 different types of Heschel's gyrus morphology in human beings as opposed to just 3. These morphological variations appear heritable, and they appear aligned to the microstructure and to functional parcellation of these regions. So thanks for the chance to chat about this. If you have questions, I guess there might be time for 1 or 2, but definitely come by the poster. It's on Thursday, and I can tell you all about Eno's really tremendous work to quantify variation in human auditory cortex anatomy.
Questions? Do you have some, really interesting, do you have some more details about the potential link to some high-level behavioral things and what you speculate is going to happen with this?
Yeah, great question. So do we think that there are different kinds of folding patterns across, for instance, developmental communication disorders or auditory expertise? This is a good question. The Human Connectome Project, unfortunately, doesn't have a lot of behavioral data associated with it, but there's other large projects out there that do, like the Adolescent Brain and Cognitive Development Study. And so we'd love to take this kind of approach to some other data sets and see what else is there. Or our colleagues in the UK can get access to the UK Biobank, which is a tremendous resource that could, answer some of these brain behavior correlations at the kind of like population-wide level. Yeah, it's a tremendous question.
Hi, thank you. One question is, how did you realign the brains to start the clustering? In principle, you have to establish a correspondence there, right? I mean,
right.
What is the consequences on what you find?
Yeah, that's a good question. Yeah, thanks for that question. We align them based on the entire brain curvature pattern using FreeSurfer. So it's a spherical alignment to the FS average template, which accounts for some of the alignment in auditory areas, but also the curvature patterns across the entire brain. So it's true that there's a big assumption there that they can be aligned in this way. But the nice thing is, if there are different patterns in the curvature, they should align to the template in a similar way. And so when we're looking at the vertex-wise distribution of curvature patterns, even transformed into the template space, those should be coherent from person to person who share an underlying curvature category.
So another question then, what you would suggest? Because everyone that has tried to map atlases to auditory cortex data knows that they just are wrong, right? I mean, most of them. So what would you suggest? Like to have different version of the atlas depending on the subgroup? Or?
I think that the long-term goal of this and related work in other brain areas is very much that. Can you identify unique patterns of curvature in the human brain and pick a better template to align them to, and then look for sort of template alignment. So can we build a template that has a complete posterior duplication? Mm-hmm. And if we identify the brain as having that sort of sulcal variation, that we could align it to that template. And certainly in terms of atlases that label cortical areas, that the functional labels would correspond better to the macroanatomy.
Shall I try?
Oh, that's so scary.
So, nice presentation. I recently came across some work of Kevin Wiener, who's at Berkeley, who you probably know has been exploring tertiary sulci and finding some extraordinary correlations with functional maps in the human brain. So I'm curious, have you played around higher up in auditory cortex to look at some of the tertiary sulci, the very small ones, that may be informative?
Oh, a really great question. No. We haven't. That would be very interesting if you think about the curvature patterns and you say, well, let's high pass filter it and get rid of these kind of large curvature and just look at the small curvature and see if it's informative. I think the end goal from all of this is definitely this kind of question: is there macrostructural functional alignment in the human brain, or does the pattern of curvature is it built around other things, right? Do functional areas emerge from the macrostructure, or do they just kind of tend to end up on the same gyrus? And there's definitely a lot of fascinating work to do here. Thanks, Jonathan.
All right. Thanks very much,
Thank you.
Tyler.